Okay, so let's get started. Good day, good evening, and welcome to all the attendees joining us from around the world. Welcome to the ARIMA HiChip webinar. Um, we are humbled and appreciative that you've decided to spend an hour with us during these uncertain and tough times. It's affecting us in different ways, and we really appreciate being here. My name is Joe. I'm Commercial Director at ARIMA Genomics, and thank you for being here. Let's take a look at the agenda. So as you see on the screen, the first speaker is our very own VP of R&D, Anthony Schmidt. And Anthony's talk is titled ARIMA HiChip, Accurate and Cost-Effective Discovery of Active Gene Regulatory Interactions. Second, we're very pleased to welcome Dr. Lorenzo Puri from the Sanford Burnham Institute. And Dr. Puri's talk is titled HiChip to Shed Light on Regulatory 3D chromatin interactions during cell lineage determination and terminal differentiation. So these should be two very good talks. We're very much looking forward to it. After each talk, we will take 5 to 10 minutes and address some of those questions. So with that, I think we're ready to end, and I'm happy to present our first speaker, Anthony Schmidt.
Thanks, Joe. I'm Anthony Schmidt. I lead R&D at ARIMA Genomics, and today, as Joe said, I'm going to be introducing ARIMA HiChip technology and its utility in identifying active gene regulatory interactions. Oops. So before we get into the nuts and bolts of the technology, I wanted to provide a little bit of introduction and motivation for studying genome organization. The genome is organized into hierarchical layers of genome organization. This is from a review from Bing Ren's lab, where I did my PhD. Starting at the highest level here, looking down at an interphase nucleus, you see the presence of chromosome territories, like you see here. That schematic is supported by chromosome painting data, as well as Hi-C data shown here on the right, where most of the interactions that you see via Hi-C are intrachromosomal interactions from chromosomes folding upon themselves in an interphase nucleus. If you go over to the left and you continue to zoom in, you'll see other structural features like organization of the chromosomes with the nuclear lamina, or presence of transcription factories more toward the interior of the nucleus. And as you zoom further in, kind of on the megabase scale, shown here, you see the presence of self-interacting chromatin called topological domains or contact domains, or TADs for short, which are demarcated by architectural proteins like CTCF, shown here. So schematically, what we're talking about here is the concept of two pieces of DNA along the linear genome A and B. And if the folding of the genome was random, you might expect some type of, crumpled up ball of yarn where A and B are also close in three-dimensional space. But what you observe from, Hi-C and other microscopy-based studies is that just because A and B are close along the linear genome doesn't mean that they're close in 3D space, and they might actually be spatially partitioned into neighboring domains, as you see here. And these types of self-interacting domains are often portrayed as triangles like this. And when you look at actual high resolution Hi-C data, this looks something like this, where you see these triangular self-interacting domains that are evidenced by higher interaction signal. And when you zoom in even further on those, you see these frequent point-to-point interactions that would be reminiscent of what's depicted over here at the highest resolution of these chromatin loops, facilitated by architectural proteins like cohesin, which have the ability to bring together cis-regulatory elements or other structural functions. So don't have a lot of time to talk about why to study the 3D genome, but broadly speaking, these types of chromosome conformation capture technologies have been used widely over the last 10 years in a variety of different biological research contexts, one of which is development. And I can just point you to a couple publications, one from Bing Ren's lab on the left, studying embryonic stem cell differentiation into different germ layers like neural progenitor cells and mesenchymal stem cells. And then on the right, a study from Giacomo Cavalli's lab looking at mouse development in vitro and in vivo. It's also been used to characterize many different diseases like Fragile X syndrome and cancer. And a couple papers there that come to mind is this paper from a couple years back from the Phillips Kremens lab at Penn, who studied short tandem repeat expansion, such as in the FMR1 gene associated with Fragile X syndrome and how that impacts genome topology and disease. And then more recently, a study from the Sirigos and Iafontis labs at NYU that was on the cover of the April issue of Nature Genetics this month, looking at genome conformation in T-cell leukemia. So as an overview, I'm going to just give another brief introduction to Hi-C to sort of set the stage for the HiChip technology that I'll discuss at greater length. So for Hi-C, I wanted to introduce the ARIMA Hi-C workflow. So for those who are already familiar with Hi-C or the ARIMA Hi-C kit, this will be background information. The Hi-C technology here is really characterized at a workflow perspective by fixing cells, just like you would for ChIP-seq, and then introducing a cocktail of restriction enzymes to fragment the genome. And now, so the blue and the gray piece are being held together in 3D space by these proteins depicted here. You digest, and then you label those ends with a biotinylated nucleotide and then ligate those ends back together. So now the blue and the gray piece that were not joined together, but held together in 3D space are ligated together with biotin marking the junction point of where those are ligated together. So then you purify out that DNA, and then we have a quantitative and predictive quality control checkpoint here. This entire process that we refer to as Hi-C can be done with our kit in approximately 6 hours. I don't have a slide for this, but we can talk about different sample input requirements if that's of interest. This process takes about 8 steps, and it happens in a single tube reaction. There's no need to wash out, lysis buffer or other things that might be prone to cell loss. We use multiple restriction enzyme chemistry, and we have these predictive QC checkpoints. After you generate this, you still have to fragment the DNA here because these molecules are too large to sequence. And then you enrich here for your ligation junctions that are informative because they contain molecules that are blue and gray and therefore are 3D interactions. And then you just carry those chimeric molecules through library prep and sequencing using a variety of different validated library prep kits. This protocol is compatible with low input. We've gone down to 50,000 cells or less, and I'm not going to show any of that data, but I will point you to a recent publication from the Akbarian lab at Mount Sinai who have used our kit for the analysis of 6,000 dopaminergic nuclei that were sorted from postmortem brain samples. So you can see that for some low input Hi-C data. And of course, we offer this as a kit and a service format. When we first started to generate these data, we compared to really the gold standard workhorse in situ Hi-C method that was developed by the Aiden lab at Baylor. The Aiden lab back in 2014, looking at in situ Hi-C data from approximately 3.6 billion raw Hi-C reads. And we wanted to compare to this gold standard data set to benchmark our Hi-C technology. And what we did was we sequenced an ARIMA Hi-C library to about 1/3 of the depth, so 1.2 billion reads. And we looked at the same exact region, and this would be characteristic of that point-to-point so-called chromatin loop that I introduced in the first set of slides. And what we see is even at 1/3 of the total depth, the signal enrichment at that structural feature that you're often doing Hi-C to detect is enriched. We see more interactions at this loop, a stronger signal relative to the local background, despite there being only 1/3 of the total number of reads. Getting at the sort of main point that I want to make here is that the underlying Hi-C technology built into the ARIMA Hi-C protocol can detect these structural features at greater signal strength despite having less reads. And what you might hypothesize is just at a genome-wide scale, if you're able to detect greater signal enrichment of these chromatin loops at individual cases, you might just be more sensitive to detect loops at a genome-wide scale. And what we did was we took the 1.2 billion reads and we subsampled that all the way down to 600 million reads, and we still identify 50% more loops than the 3.6 billion read in situ Hi-C data. And our customers have also been able to experience this as well. This is analyzed customer data in IMR 90 cells, driving at that point of being able to detect more loops from less sequencing. That's all I'm going to say about Hi-C. You can look at our website for testimonials for those who have used our kit, like Eileen Furlong at EMBL, Toshi Shioda at Harvard, and Hirwe Meharana at MIT, who's been using this for low input and the analysis of precious rare clinical sample types. Our website also has a list of publications and preprints in this field. And with that, you know, I would just say that as a company, we've been really pleased with the performance of the underlying Hi-C chemistry and the Hi-C platform. And we're trying to expand that to other targeted genome confirmation assays because we realize that certain biological questions are better suited for targeted genome confirmation approaches, such as Capture Hi-C or HiChip. And the focus of this will be on HiChip as a protein-associated genome confirmation tool, which I'll describe in greater detail now. So shifting to the second part of the presentation, we'll focus on HiChip. Now, first and foremost, simply what is HiChip? So for those who are not as familiar with HiChip, it may also be called PlaqueSeq. Those are effectively the same technologies, just with two different terms. HiChip was first published by Howard Chang at Stanford back in 2016. And then just a couple months later, a very similar technology called PlaqueSeq came out of Bing Ren's lab. It was one of the final things that I did when I was in Bing's lab, and it was great to get that hands-on experience with the technology before moving to ARIMA. And both of these are simply characterized by in situ Hi-C combined with ChIP-seq. So to dive into the nuts and bolts of that workflow a little more, so for those who are familiar with doing ChIP-seq, it's nice to frame HiChip relative to a ChIP-seq assay. So for ChIP-seq, you'll start with your fixed cells, just like I've shown before, and you have your cross-linked DNA. That chromatin effectively gets sheared, and then you bind with your antibody, and you do your IP. And then you carry through that immunoprecipitated DNA into library prep and sequencing. The whole thing might take a day and a half or 2 days. HiChip, in our implementation of HiChip, which is sort of the classic in situ Hi-C plus ChIP-seq, is really just effectively the ARIMA Hi-C chemistry to generate the proximally ligated DNA. So the high fidelity capture of 3D interactions first. And then when you get to this point where you have your proximally ligated chromatin before reverse cross-linking, you just shear and you add your antibody and you do your IP. And the difference you can see is that what you are enriching still has the sequence content directly next to the immunoprecipitated protein, but these molecules also contain 3D interaction information here because the input is proximally ligated chromatin, not genomic DNA chromatin. Right. So then you just carry through and you enrich for the ligation junctions and then you sequence. So what's sequenced are effectively these molecules that were immunoprecipitated and associated with that target protein of interest. Right. So this technology does inherently capture the 1D protein occupancy or protein binding sites that you would get from ChIP, but also on top of that, the 3D interactions that were associated with that immunoprecipitated protein. And what most of the customers that we've been working with so far are using this technology to characterize something like H3K4 trimethylation and to extract the chromatin loops that are associated with, let's say, active promoters. We have optimized sharing protocols with Diagenode and Covaris instruments, and we did a collaborative poster with Covaris this past year at the AGBT conference. And these are also offered as kits or services. So why use HiChip? It's a pretty new technology with not nearly as many publications as Hi-C, but there has been use of HiChip across a variety of different research areas. For example, Brad Bernstein's lab has used HiChip to analyze the topology of gastrointestinal stromal tumor sample genomes. And Paul Mitchell's lab has characterized circular extrachromosomal DNA using HiChip. There's another, I would say, bucket of applications where you might want to identify enhancers that are in GWAS regions and use something like K27 acetylation HiChip to link those enhancers in the GWAS regions to putative target genes that may be involved in disease pathogenesis. And these studies have also used comparisons to CRISPR-I data to identify that the HiChip loops that you're finding are functional because there are cases where they're supported by CRISPR-I experiments, such as at the MYC locus in the paper from the Chang lab at the top there. But also other applications like development. Here's a study from Effie Apostolou's lab at Cornell, who's used HiChip to study sort of a protein-centric view of genome organization during cellular reprogramming, and really can be applied to any transcription factor-driven process. We've spent quite a bit of time during product development looking at open source bioinformatics pipelines for the analysis of HiChip data, and we've tested several pipelines, and we do support several, but we recommend the MAPS pipeline that was developed by Ming Hu's lab at the Cleveland Clinic. So we've been having a nice collaboration with Ming to develop more user-friendly reporting features that are output from this pipeline. And the way that it works is you just take your FASTQ files that you get from the ARIMA HiChip data, you feed those FASTQ files into an alignment program called Feather. Those mapped HiChip reads get put into a statistical framework called MAPS, which is what calls the statistically significant loops from the HiChip data. And what we've done is built on top of this pipeline, the standard output will be these arcplot files that I'm showing here that represent all the statistically significant chromatin loops. And you can upload these directly into the WashU genome browser to view the chromatin loops relative to the other 1D tracks like RNA-seq or ChIP-seq or open chromatin assays. By default, this pipeline also produces a quality control table that has high-level key performance sort of metrics like how well did the chip work and how well did the Hi-C part work of the HiChip assay. And then importantly, if you have pre-existing ChIP-seq peaks, we will create these meta plots and heat maps, which effectively just pile up all of the HiChip reads relative to the known chip peaks. And if the chip enrichment worked, which is really critical to look at, you should see an enrichment of the HiChip reads relative to the known chip peaks. And when we look at some of our internally generated ARIMA HiChip data, and how does that compare to the enrichment that you would get from ChIP-seq assays? Well, here's a metaplot of GM12878 H3K4 trimethylation ChIP-seq data that was downloaded from ENCODE. So here you see about a 6-to-1 peak to local background enrichment. And when you plot the ARIMA HiChip data the same way, using the same protein factor, you also see about a 6-to-1 peak to background enrichment. So quantitatively, also in K27 acetylation, it looks very similar. You see very similar genome-wide chip enrichment profiles in the HiChip data compared to the ChIP-seq data. Now, what does this data look like? What I'm showing here now is a snapshot of the MYC locus and several different publicly available ChIP-seq or RNA-seq based tracks in GM12878 cells. What I want to draw your attention to here is the bottom two tracks, which are K27 acetylation ChIP-seq data. Now, if you plot the HiChip data just like ChIP-seq data, which I'm now showing here, you can see that the chip profiles look very similar to what you would get from ChIP-seq, and that's just, of course, a good sanity check that the enrichment worked, like I showed on the last slide. But of course, what you're not getting from ChIP-seq is the 3D connectivity information that I'm now layering on here that you get also from the HiChIP data, right? So it's sort of like two different data, features in one. And for example, here's the MYC gene right here at this CTCF site. And you can see loops to these very distal K27 acetylation peaks and lots of other connectivity here. Now, we were pretty thoughtful about trying to ensure that the loops, that we are calling are reproducible between replicates. And, qualitatively, they look really similar here. And when you quantitate that at a genome-wide scale, what we observe is that for K27 acetylation HiChIP, approximately 85% of the loops that are found in one sample are also found in the other replicate. And when you look at our K4 trimethylation HiChIP, the replicate concordance is somewhere between 74 to 87%. Right, so on average, approximately 80% of the loops found in one replicate are also found in the other replicate. Now, I really want to stress the point that we've done quite a bit of customer validation of the HiChIP protocol, and I want to talk through this from the beta testing program that we ran. And the way I think to frame this discussion is to talk about what are some of the key performance metrics that you would want to think about when it comes to HiChIP data. And those two top level metrics really are, well, how efficient does the HiChIP protocol capture the 3D aspect of genome conformation? How efficient was the proximity ligation portion, or the Hi-C? And then how specific was the chip enrichment? Okay. Now, what I'm plotting here is the fraction of the HiChIP reads that overlap known chip peaks. This is very much like the FRIP metric when it comes to ChIP-seq. And then what fraction of the reads represent long-range chromatin interactions? And of course, you want to be in the upper right quadrant where you have a high fraction of chromatin interactions and a high degree of chip enrichment. And for all 3 beta testing sites and all 24 reactions encompassing their own supplied samples as well as some controls, you see a high degree of chip enrichment and long-range cis contacts. And what this translates to when you have a high degree of chip enrichment and long-range contacts is it drives down the cost of sequencing to identify loops associated with active chromatin features. So here what I'm plotting is the fraction, is the raw reads that were required for reproducible loops, 3 different beta testing sites, and then different cell types that were analyzed across the x-axis here. And the overarching theme here is that for K4 trimethylation HiChIP, most of the beta testers needed between 100 and 200 million reads. And then for the K27 acetylation, which is in gray, most beta testers needed somewhere between 200 million or 350 million, somewhere in that range. Now, to just broadly take a step back and compare that to Hi-C, I believe what ENCODE is recommending for in situ Hi-C is approximately 2 billion reads per sample. I showed you in the Hi-C introduction that for ARIMA Hi-C, we recommend at least 600 million reads per sample. And then with HiChIP, because of that enrichment, you're driving down that sequencing requirement significantly to somewhere in the 300 million range for K27 acetylation, or somewhere around 100 million reads for K4 trimethylation. And lastly, these types of data can be used for differential analysis. So what I'm showing here at the top is a set of loops that are anchored at the promoter of a gene called ITGB1BP1. And in the lymphoblast cells, the majority of those interactions are actually occurring downstream to other H3K27 acetylation sites, with, I guess, one exception of this upstream interaction that's going to this CTCF ChIP-seq peak over here. Now, when you compare this to a sample like iPSC-derived neurons, this is data that was generated by Yin Shen's lab at UCSF, the significant interactions are dramatically skewed upstream, right? even though there are K27 acetylation peaks downstream. So you would have not known these types of interactions if it weren't for the HiChip data. And what you can see is only the neuron samples have a CTCF peak right here at the promoter, and perhaps that's kind of confining the, interaction landscape to this area, whereas the lymphoblast cells don't have that CTCF peak, so perhaps it has the spatial flexibility to interact both downstream and upstream. And these are just the types of analyses that you could envision doing with the HiChip data. We have quite a few customer testimonials now, including one from the next speaker across different research areas. Peter Scacheri's use of the HiChip data in cancer research, as well as Tomas Ordog at Mayo and Yin Shen at UCSF, who got the HiChip to work on samples that were previously very challenging with homebrew HiChip protocols. So in conclusion, I hope I've spoken to the streamlined in situ HiChip workflow that we have for efficient capture of both 3D loops and 1D chromatin binding profiles. I showed you that the reproducibility between loops, between replicates, is about 80% using the HiChip protocol. We have extensive customer validation across different protein factors and cell types, and importantly, an end-to-end open source bioinformatics pipeline, and we do significant technical onboarding so that you're able to analyze your HiChip data. So in addition to our beta testers and partners really throughout this entire product development program, I want to thank our scientific advisors, Jaap Dekker, and our two co-founders, Jesse Dixon and Bing Ren, as well as the very generous funding support we've received from the NIH since the company's inception. And with that, I'm happy to take any questions.
Fantastic, Anthony. Thanks. This is Joe again. And we've had some very good interaction throughout your talk. So very excited to ask some of these. Listen, there was numerous questions around sample types as well as input requirements for both Hi-C and HiChip. Can you comment on that, please?
Yes, for Hi-C, we have validated protocols and user guides for animal tissues, both vertebrates and invertebrates, primary cells. I showed that one slide there that had the post-mortem brain samples, of course, cell lines, plants. And for Hi-C, the input requirements, we typically recommend at least 50,000 cells, even though the Akbarian lab could push that down to just over 5,000. For HiChip, I think I have a slide in the backup somewhere here. Yeah, here are some of the examples of sample types that we have validated with the ARIMA HiChip kit. So cells like lymphoblast, fibroblast, iPSC-derived neurons, gastrointestinal stromal tumor cells, liver cancer cells, lymphoma cells, glioblastoma cells, CD8 and CD4 positive T cells. We've worked with in vitro differentiated Th1 and Th17 cells. And then one of our customers has also used the HiChip protocol for the analysis of heart tissue. And I believe that workflow involved isolating nuclei from the tissue and then putting the nuclei into the HiChip protocol. So those are the different sample types. We typically recommend, I believe it's approximately 3 million cells per reaction would be the ideal amount. Somewhere in the range of, let's say, about 12 micrograms of chromatin within the cells that you're putting into the HiChip assay. And you derive the optimal cell count empirically before you start HiChip using our kit. So whatever cell count 12 micrograms of chromatin translates to is what you'll use for HiChip.
Great, fantastic. Another round of questions regarding validation with the histones that you showed, H3K27 acetylation and H3K4 trimethyl.
Yep.
Many questions about transcription factor. Can you comment about HiChip for transcription factors?
Of course, yeah. So over the course of the past year, when we've been developing this technology and bringing it to market, we've focused majority on histone mods, particularly those two that I've presented today. But really the goal is for this to be an open platform to do several different chromatin binding proteins, or TFs. We have done a little bit of work with a cohesin subunit as well as CTCF. And we're rolling into phase 2 now of the technology development, where we're going to do more internal and extensive validation with other transcription factors. And then I should also say that we already have several customers who are just going ahead and using the kit for transcription factors with the understanding that if it's a well-performing antibody, that the technology should work. So we have people that are just plugging away with TFs on their own with our extensive support through our tech support team.
Great, great. Final question, and then we'll move
Sure.
on to Lorenzo. There's a couple questions. One, does Hi-C and HiChip kit reagents contain exactly the same chemistry, same components and concentrations? And two, between Covaris and Diagenode sonication instruments, which one is superior or easier to work with and optimize? Thank you.
Sure. I'll do the second question first. So internally, yeah, we have both instruments and we've used both. I would say in general, we get higher chip specificity by using the Covaris, and we have both the Covaris parameters as well as the Diagenode Bioruptor Pico parameters in our user guide. We have seen that the Covaris has been pretty robust as long as you follow our cross-linking protocol across different cell types. So for all of the beta testers, they all used a Covaris and just used our recommended power settings, and all of that data that I showed that worked on the first try was using the Covaris system. Regarding the question about the Hi-C kit components and the HiChip kit components, our latest kit, which we're calling Hi-C, ARIMA Hi-C Plus, has all of the reagents that you would need to either carry out a Hi-C reaction or carry out a HiChip reaction, or capture Hi-C for that matter as well. But it's effectively one kit that comes with 8 reactions, and you can use those 8 reactions for however you want, whether it's 4 Hi-C and 4 HiChip, but all those reagents are there to carry out either 3 of the protocols that I talked about today.
Great, wonderful. Thanks, Anthony. And
Sure.
let's move to Lorenzo. And just so everyone knows, we didn't get to every question. However, we are logging these, and we will answer your question at the webinar if we don't get to them during the webinar. Lorenzo, welcome.
Thank you. So we're thankful to ARIMA for involving us in this operation. So we're very excited to use HiChip in our system. This is a HiChip protocol with antibody against H3K27 acetylated moiety. And we have applied this HiChip protocol to our favorite setting. This is the typical myogenic conversion of fibroblast by the ectopic expression of MyoD, the myogenic lineage determination factor. This has been a seminal discovery from Weintraub lab, from Weintraub and Lassar, and has been followed up by illuminating work of Steve Tapscott lab. So why is this a very good system suitable to understand the role of H3K27 acetylation marked chromatin interaction? First of all, because this is a very highly regulated and reproducible experimental system that synchronizes cells at different stage of transdifferentiation from fibroblast to mature differentiated multinucleated myotubes. It also provides one reference transcription factor, which is MyoD, that helps to match iChip, IC, and ChIP-seq profiles through the genome and provide a general picture of how the transcript factor that is overexpressed, or actually in this case is epically expressed, these interactions and with respect with the acetylation of chromatin. It also provides a parallel stage-specific gene expression profile, and more importantly, as emphasized by this slide, this system is operated through the ectopic expression of only one transcription factor, as MyRD is unique among all transcription factors for its ability to promote conversion of somatic cell types into skeletal muscles by the ectopic expression without requirement of ectopic expression of other transcription factors. The other good advantage of using MyoD-mediated myogenic conversion is that MyoD is well known as one of the first lineage determination transcription factors that's been associated functionally and physically with the direct the enzymes that are directly implicated in promoting histone acetylation or removing the acetyl radicals from histones. And as a matter of fact, works from Tapscott lab has shown that the ChIP-seq chromatin binding profile of MyoD is highly associated with local histone hyperacetylation. Finally, from a biological standpoint, we know that the expression of MyoD is a fundamental event for the commitment of muscle stem cells into the myogenic lineage. So we know that also in vivo during adult skeletal myogenesis, which is essentially muscle regeneration, MyoD expression turns skeletal muscle stem cells into myogenic committed progenitors and later on multinucleated myofibers. More importantly, we know that epigenetic drugs that impact on histone acetylation like HDAC inhibitors and BET inhibitors can modulate skeletal myogenesis in a stage-specific manner, and we also know that some of these compounds are currently used in clinical trials to promote muscle regeneration. This is our model. As you can see, MyoD is uniformly expressed upon the doxycycline exposure in fibroblast. This is a system where there is a doxycycline regulated expression of MyoD. You see that the large majority of the fibroblasts express MyoD, and at this stage, we have RNA-seq. You can appreciate that at this stage something very interesting happens, and this is the expression of myogenic factors and the consensual erasure or repression of a lot of markers of fibroblastic cell line. And later on, this results in the formation of multinucleated myotubes. So what we have learned using this system is that by combining IC and ChIP-seq, and RNA-seq, of course, is that MyoD operates to promote conversion of fibroblasts into myogenic cells by binding chromatin loops. As Anthony already explained in the previous talk, these loops are organized such that there are two anchors or boundaries which are co-bound by CTCF and cohesion, and that insulate chromatin interactions that refers to promoter and other regulatory elements like enhancers. Looping. So we have learned that MyoD can rewire or even change the interactome within these loops by promoting new promoter enhancer interactions, and at the same time MyoD binding proximity of CTCF at the anchors or boundaries of this loop generate changes in the strength of interaction that regulate the gene expression from the boundaries. So here are two paradigmatic examples. This is a typical genes where MyoD binding that you can spot here at the promoter region of this troponin can instigate a binding which is verified by 3C experiment with a distal enhancer located intragenic in a gene within the same loop where MyoD binds. And here instead is alpha-7 integrin, and you can appreciate that MyoD binding at this time is in a close proximity with CTCF, which reflects the boundaries of this loop, the anchors, and this binding results in the increased trend of interaction between these boundaries and the consensual increase in gene expression of the genes within. There are in the loop defined by these anchors. Still, we realize that the precise relationship between the binding profile of MyoD, its transcriptional activity, and histone acetylation, and the related change of chromatin, three-dimensional chromatin loops was still incomplete, and we decided to address this with iChip using the ARIMA kit. As already shown by Anthony, our samples were within the same range of the other beta tester samples in terms of percentage of captured long-range interactions and the percentage of overlapping between iChip reads and ChIP-seq peaks with the same antibody, which was H3 K27 acetylation. Here is the relative enrichment of our high ChIP signal related to the ChIP-seq profile with the same antibody, and you can see that the enrichment shows a very, very good overlap in both fibroblast and MyoD committed fibroblasts into the myogenic lineage that we refer as a myoblast. And there was also a very good replicate overlap ranging around 80% of the common interactions that we capture with the iChip in the two replicates, biological replicates of our fibroblast. I should actually point that those are human Imer90 fibroblasts, just to be precise. And the same, we found a consensual 80% reproducibility when we use the iChip with MyoD converted fibroblast into the myogenic lineage. Here is one data that, for instance, differs iChip with this antibody with the data that we obtained by using IC and the ChIP-seq, which is immediately portrayed by this graph. As you can see, here we have all the interactions captured by iChip that involves promoter, light, and dark blue, other distal non-promoter elements, which are far from the space 1.2 kb and plus 5.5 kb from the transcription site of genes within the human genome. As you can see, there is a partitioning of iChip signal in fibroblasts between these two categories of genomic elements. And as you can see, upon the introduction of MyoD, there was a clear restriction, compression of the elements that interacts, which suggests what MyoD indeed does, which is restricting the repertoire of genes that need to be activated. So when we look at the MyoD-bound interactions captured with the H3K27 acetyl antibody, we found that most of the interactions that we captured that involved promoters were also bound by MyoD, while only 40% of the interaction captured outside of the promoters were bound by MyoD. Those interactions are likely to be enriched with enhancers, and we conceived that probably super enhancers were important for the myogenic lineage commitment driven by MyoD. So we initially decided to use the computational prediction of super enhancers in our system, which is MyoD-mediated commitment of fibroblasts into myoblasts. And we found that 1,343 super enhancers could be actually predicted by ChIP-seq, a profile with H3K27 acetyl antibody. And when we looked at the super enhancers that we could detect with the addition of one of the most important features of super enhancers, which is their ability to interact, 72% of these super enhancers were detected by iChip within the other, which are enriched in the enhancers, so it makes absolutely sense. And 80% were detected within the promoter regions. I should say that sometimes, because of the different binning, in the same bin, 5kb bin, we can find the promoter and part of an enhancer, which is called, therefore, within the promoters. So overall, 90% of the predicted super enhancers could be actually captured by IC, which I believe includes a criteria of specificity provided by the potential interactions between super enhancers. We also looked at the ChIP-seq, iChip profiles and the reproducibility of the interaction captured in the fibroblast and the MyoD ectopic expressing fibroblast, which we call myoblast. And you can see that there are pretty reproducible interactions captured in the fibroblast, also in the myoblast. When we look at all the interactions, either induced or downregulated in MyoD expressing fibroblast, we found that those interactions that were increased usually correlate with a better binding of MyoD. So most of the MyoD peaks, by looking at the ChIP-seq profile of MyoD, were found within interaction captured by iChip interaction that were increased in their strength, while those interactions whose strength was decreased were still associated with MyoD binding, but at much less extent. And here's this representation, and you can see here a very interesting feature that could be revealed only by iChip. We could not come up with this immediate picture by integrating IC and ChIP-seq profile. You see here related of MyoD and that were increased in their strength showed a consensual increase in the H3K27 acetylation. So this clearly associates MyoD binding, increased interaction strengths, and histone acetylation, while a lot of the large majority of interactions whose strength is actually decreased upon ectopic expression of MyoD could still be bound by MyoD, yet the acetylation did not increase. In terms of biological readout, this has a meaning as shown here, because all the increased interactions tend to correlate with the activation of genes implicated in skeletal myogenesis, while most of the interactions whose strength was decreased upon MyoD ectopic expression were associated with the decreased expression of genes that refers to the lineage, fibroblastic lineage. And I should note that most of these pathways are actually anti-myogenic. So it makes absolutely sense that MyoD disarm or downregulates all these pathways or genes networks. And here, I want to show you some examples of how the iChip can visualize all the interactions marked by H3K27 acetylation that are instigated by MyoD. So this is the first example. I'll go through a few upregulated and some downregulated genes. So this is an actin that is upregulated by MyoD. You can see that this is the gene, as you can see by RNA-seq upon ectopic expression of MyoD, there is a clear induction of this gene. And you appreciate that in fibroblasts where MyoD is not expressed, there is a clear interaction between these regions, which appears probably close to the promoter, but I don't believe it's part of this promoter, and promotes interactions with different distal regulatory elements that are likely enhancers. Upon expression of MyoD and binding to the promoter of this gene, which is shown by my arrow, you appreciate that now there is a new acetylation that is clearly associated, whose increase is clearly associated with MyoD binding. And these events is sufficient to instigate a huge number of interactions, especially with other MyoD-bound distal regulatory elements, which becomes also hyperacetylated. So this is a typical disengagement, simultaneous disengagement of previous interactions, as you can see here, and refocus on MyoD-binding chromatin elements that get hyperacetylated and establish promoter enhancer interactions. This is instead a beautiful example of muscle-specific genes whose expression is a dysferlin, is appreciated by RNA-seq here, is induced by the ectopic expression of MyoD, and this is a very beautiful example of de novo interaction established by MyoD binding at the promoter of this gene with other MyoD-bound elements, which are likely to be MyoD-induced enhancers or MyoD-activated enhancers. And this is a paradigmatic example of how iChip with H3K27 acetylation antibody can actually reveal interactions between promoter of a MyoD target gene and distal elements that were not captured previously by our combined IC and ChIP-seq analysis. So the interaction that I've shown you in the slides during the presentation was related to this promoter bound MyoD and this intragenic sequence within this gene called LAD1. And that was the only interaction that we captured with our previous analysis, IC ChIP-seq integration. But as you can see here, with iChip, we found a huge number of interactions instigated by MyoD binding to the promoter. And that revealed a huge number of additional putative enhancers that completely escaped our attention, from our attention in our previous analysis. And finally, I want to show you how this iChip kit can reveal a beautiful feature that we're actively investigating and refers to the MyoD ability to repress gene expression through the disengagement of previous interactions in fibroblasts. You see here a case where MyoD binds the promoter of a gene, which is downregulated. It's kind of paradoxical that a transcription activator downregulates the gene by binding the promoter. But you see here, we are trying to understand why that happened. MyoD binds a promoter that is also bound by CTCF, This could be a boundary of one of these loops. As a matter of fact, the binding of MyoD results in the hypoacetylation of most of the hyperacetylated elements that are supposed to control probably the expression of this gene, and this results in the disengagement of most of these interactions in MyoD expressing cells. This one is even a more drastic phenomenon of disengagement of multiloops are supposed to drive the expression of a gene that you don't want to see expressed in skeletal muscle cells because it's GATA6. It's a gene that drives lung differentiation. Those are lung fibroblasts. It makes sense that this gene is expressed in fibroblasts, but as a matter of fact, as soon as MyoD lands on the chromatin, you see that all the MyoD binding correlates with disengagement of a huge number of multiloops, and this promotes the repression of this gene. So with the RIMA iChip kit, we found a very easy to use, optimized kit protocol that was working perfectly in our system. It could capture highly reproducible chromatin loops, which are marked by specific histone modification like H3K27 acetylation, and that whose detection typically requires the combined and correlative analysis of IC and the ChIP-seq data. So with the iChip, we can reduce the sequencing cost and capture a high resolution data. Is a suitable approach to capture chromatin loops between active regulatory elements that drives specific biological programs instigated by transcription factors. And we believe that this is probably one of the best approaches to monitor the effect of certain epigenetic drugs that target the post-translational modifications. In this case, were histone modifications. So it seems to be a suitable system to capture modifications from epigenetic drugs like HDAC inhibitors and BET inhibitors. Let me just thank the 3 people that have done this analysis. Luca Caputo is the postdoc who made all the iChip experiments using the RIMA kit. Chiara Nicoletti is a bioinformatic postdoc that analyzed the data, and Alessandra Dalagnese is a former PhD student who is now postdoc at MIT in Rick Young Lab, who initiated the whole project by providing the all IC and ChIP-seq dataset. Thank you guys for your attention.
Great, thanks, Lorenzo. Got a couple of questions for you here. I know we're running really low on time. Let's start with maybe the second one, which is, it seems like the next step would be to look at HiChip using something like MyoD. And do you plan on doing this, and what would you expect, and how would it add to your existing K27 acetylation HiChip data?
So yeah, I mean, it's clear that the optimal experimental setting would be to match iChip profile of histone modifications and iChip profile of transcription factors like MyoD. We are clearly working in this direction, and I believe that probably even a more interesting setting would be to move into in vivo experiments. So the effort is to reduce the number of cells necessary for this analysis and start to work with FACS isolated cells using 2 settings of cell types that express and do not express MyoD, which are actually quiescent, for instance, skeletal muscle stem cells, and myogenic progenitors, which are activated upon injury, which is our setting to promote muscle regeneration.
All right. And one other one was, um, I think it was regarding the evidence that you showed by the gene ontology analyses. Have you done any other orthogonal validation of your loops using non-3C based approaches?
Yeah, can you hear me?
Yes.
Yeah, I mean, this is published in our molecular cell paper that came out in November. So we have used and we continue to use several orthogonal validations. They may range from orthogonal analysis like Fish and 3C analysis, but also functional validation with that Cas9. So with CRISPR-Cas9, we could show that most of the MyoD loops that result in the activation or repression of gene expression can be abrogated by eliminating the MyoD chromatin binding with that Cas9. So I think it's absolutely necessary to use this orthogonal validation. But more importantly, when we have done that, that validated our iChip data set.
Okay, we have a question also that I think is addressing the ACTN1 gene example, where I guess the question is there's not a lot of direct interactions in the myoblast at the promoter, but you can see that there's sort of just an increase in interactions just around the gene. Do you think that direct interaction with the promoter is necessary, or just broadly around the gene itself?
We do believe that we have to believe that this is the only perturbation in these cells. The only perturbation is the expression of MyoD and this binding of MyoD. Actually, of course, there is a simultaneous binding of MyoD to these enhancers, which leads to the hyperacetylation and therefore the enhancer activation. So you see that these two binding correlate with a clear increase in interaction between this promoter and these enhancers. Then I would like to drive the attention that these result in the further multi-loops interaction. But I think the first event that triggers the other interactions is this MyoD binding to this promoter. It is also possible that other than a typical MyoD binding, promoter binding, this could also be one of the MyoD binding to a promoter that is also part of a potential boundary, which is CTCF bound anchors of this loop. So this could simultaneously change the interaction strength of these boundaries and generate these loops. We still don't know how all these loops are generated, but clearly they refer to initial binding of MyoD to this element, this promoter, and perhaps the simultaneous binding to other regulatory elements.
All right, and lastly, have you looked at CBP P300 binding and how it correlates with MyoD binding at loci where you lose high chip interactions?
No, not yet, but we're running these experiments. We're trying to combine profiles of ChIP-seq of histone acetyltransferases and deacetylases, and then look at the consensual binding with MyoD and loop formed by potential regulatory elements that are enriched with histone acetylation. And of course, we're very interested in testing this setting to understand whether we can generate reliable readouts in terms of chromatin loops after treatment with epigenetic drugs like histone deacetylase inhibitors.
Okay, I think that's all the time we have for questions right now. Joe, I'll hand it over to you to wrap up.
Well, thank you so much, Anthony, and thank you, Lorenzo.